Well, it's really wonderful to be here. My first love in school, academically at least, was physiology. And so to be asked to speak to a group of physiologists really means a lot to me. I've also loved Irish history and Irish literature, so this is a wonderful opportunity for me to combine two passions I've carried to the present. And I'd also like to thank everyone for being so welcoming, especially Garrick and Steve, who did the best job setting up my computer of any individuals with whom I've ever interacted. Okay, title here has changed a little bit compared to what you saw in the program, and it now reads Leptin and the Remote Control of CNS Circuits that Control Food Intake and Metabolism. And I'd like to discuss in turn three topics. First, I'll just review a little bit about leptin physiology and then update you on its uses as therapeutic, as potential therapeutic for obesity and its currently approved utility as a therapeutic for a rare form of diabetes known as lipodystrophy. I'll then segue into how this work led us to an interest in exploring how the CNS controls glucose metabolism, and then transition to a description of a new method we've developed to probe this, that we call this method NICER, which stands for non-invasive cell regulation. It's a way to control neural activity using electromagnetic fields. Now, the clinical problem we study is illustrated here. The question that I suppose everyone asks themselves, scientists and lay public alike, is that if you see two individuals with this disparate appearance, you'd wonder why the difference. Now, in the lay public, at least, there are three general explanations for why weight could be so divergent among different members of our population. The first suggests that the obese lack a level of willpower that's normally exhibited by lean people. I find that point of view more often favored by lean people. A second possibility is that we live in a modern lifestyle in which activity is de-emphasized, food is readily available, and that suggestion is that this is a disease of modernity. And then the third possibility is that differences in body weight are accounted for by biological factors, genetic variation that accounts for the extremes of phenotype that you can see. Now, interestingly, in this area, there's often debate of one of these possibilities at the expense of the other. I'd like to suggest, though, that if we were talking about any other condition, cancer, heart disease, hypertension, people would immediately gravitate to the idea that there's a biological framework on which environmental and behavioral factors act. And for reasons that I think are more sociologic than scientific, debate on this topic has tended to focus on one of these possible contributors to the exclusion of the other two. And I think what I'd like to at least ask you to contemplate today is the possibility that obesity is at its core a biological disorder, a physiologic disorder, and one that in time will be treated in the same way that we treat endocrine disorders. We're not there yet, but I'd like to think that that's where the field is heading. Now, to illustrate some of these points in more detail, I want to return to one of the images I showed you. This is the young boy who was pictured on the left side of the slide that I showed you. He was first identified in Cambridge, England by Stephen O'Reilly and his colleagues. This child was of normal weight at birth, but developed morbid obesity pretty much beginning in infancy. He was markedly hyperphagic at a young age. There was nothing the parents could do, it would appear, to restrain his appetite. He was already pre-diabetic at the age of 4 when he weighed 90 pounds with 57% body fat. Now, this child came from a highly inbred pedigree, and he had a similarly affected 9-year-old cousin who weighed over 200 pounds. When you see an extreme phenotype in an inbred family, it's often the result of Mendelian defects. And so O'Reilly considered the possibility that this child might have a defect in the same gene as we had identified in mice. This is a picture of the Obi mouse identified at the Jackson Laboratory in 1951 by George Snell and colleagues. And the only difference between the mouse at the far right of the screen and its sibling littermate adjacent to him is a defect in a single gene, a single fully penetrant autosome recessive gene, the consequence of which is an animal that overeats voraciously, has 5-fold increase of body fat as part of a very complex syndrome that was curious at the time and about which I'll say more in a minute. And by this I mean this mouse also manifests a set of abnormalities that you don't normally see in human obesity. The mouse is hypothermic, the mouse is infertile, the mouse has pervasive immune abnormalities as well as some thyroid abnormalities. So they're reproductive and pan-endocrine abnormalities. In fact, many people had suggested that the phenotype of this mouse would not be instructive for our understanding of human obesity because these associated abnormalities are so different than are typically seen in obese humans. I'll try to explain that basis for this a little bit later. In any case, in 1994, we identified the defective gene in these animals as encoding the hormone leptin. And so O'Reilly quickly looked in the plasma of the young boy that I showed you an image of and found that the child did not produce any leptin whatsoever, was leptin deficient, and had a mutation in the gene. So they, as we had previously done in mice, they began to give the child leptin, and here's what happened. The child got older. That has nothing to do with leptin. That's just the way things are. But despite getting taller, he became thinner, less diabetic, less insulin resistance, I should say, with a marked reduction in body fat over the first 2 years of therapy. Before the first dose of leptin, the child was given a test meal, and at that test meal, the child ate at a single sitting 2,000 calories, which is the normal intake over a day for an adult of my size. This child ate in a single sitting and asked for more. After a few leptin injections, now the child ate 180 calories, which is the age-appropriate number of calories of a child of his size at such a test meal. Now, this is a picture of the child at 8 years of age. You'll note these are the two images I began by showing you. So in this case, the cause of the obesity was not a lack of willpower or an environmental factor. Rather, it was a lack of the hormone leptin. Now, this is an important study because it shows or confirms the physiologic role for leptin in human, but it does not, on the other hand, explain obesity in the general population because leptin mutations of this sort are very rare. The same is also the case for most all other hormones as well. And so while leptin deficiency is a rare cause of obesity and establishes a role for leptin in human physiology, it does not, however, instruct us as to what the molecular causes of obesity are. And I'll say a bit about that in a moment because a description of the identification of downstream mediators of leptin action has now led to an evolving and growing understanding of what some of the molecular alterations are that do in fact cause human obesity. Okay, so leptin is an afferent signal in the negative feedback loop that maintains homeostatic control of fat mass. Leptin is made by fat in proportion to its mass. As fat mass increases, leptin level increases. It then acts on brain centers that control appetite to reduce appetite. Conversely, if fat mass falls, is lowered, leptin level falls. The new low leptin level is a stimulus to eat more. And by this mechanism, body weight can be maintained within a relatively narrow range. So in a sense, what this defines is an endocrine circuit, the purpose of which is to maintain fat mass within a relatively narrow range. It allows you to maintain stable weight within that range. I won't have time to go into the details of this, but there's very good reason to believe that this system evolved as part of intense evolutionary pressures among mammals and other species as well, including birds, that acts to resist the potential damaging sequelae of being too thin or too fat. If you're too thin, you're at greater risk for starvation. If you're too fat, you're at greater risk of predation, of being victimized by a predator. And so this system evolved to balance those two relative risks. Now, leptin deficiency causes obesity because this factor, which normally signals that their adequate fat stores are missing, is absent. In the absence of this signal, the organism thinks it's starving, and as a consequence eats voraciously to try to restore fat mass, the consequence of which, in this case, with food being available, is obesity. Now, I mentioned that leptin mutations and leptin deficiency is quite rare. In fact, if we look at obese and lean patients, what we find is that among obese patients, actually the levels of leptin are elevated. So what this slide simply shows is the correlation between leptin concentration and percent body fat performed with Eric Ravison and Margarita Maffei. And what you'll see is about a 0.9 correlation between leptin level and fat mass. So leaner people tend to have lower levels of leptin, more obese people have higher levels of leptin. Now, when an individual has a high level of hormone, in this case an obese individual, shown on the upper right part of this curve, in the absence of the hormone effect, the effect being to induce leanness, it's generally the case that this is the result of a hormone resistance syndrome. So the best analogy of this would be insulin and type 2 diabetes, where 90% of type 2 diabetic patients actually are hyperinsulinemic. So this and other evidence I won't have time to share with you has led to the conclusion that most obesity is the result of some degree of leptin resistance. Some block in leptin action, mechanism not entirely known. And the consequence of this would be as follows. If fat made leptin and the leptin didn't work as well at its site of action, less signal would get through, food intake would increase, fat mass would increase, and you now re-equilibrate the system with higher fat mass and higher leptin levels. I'll say a bit about the pathogenesis of leptin resistance in a moment, but first I want to treat the empiric question of what happens now if you give extra leptin to patients who are leptin resistant. Will they lose weight? And the answer in the rest of endocrinology is this. If you're dealing with the hormone resistance syndrome and give extra hormone, sometimes the hormone works, albeit with reduced potency. That's the case for insulin. But sometimes it doesn't work at all. And that's, say, the case for EPO or, to a lesser extent, growth hormone. So the question was, what would giving extra leptin do for patients who already had high endogenous levels of it? Now, the first experiment of this sort that was done was published in 1999. It was a small study. And what you can see is that at the highest dose of leptin, on the far right, 0.3 milligrams per kilogram, the response appeared to be variable. 3 patients didn't respond at all, but 5 were thought to respond pretty well. This was a statistically significant effect, and at the time was considered mildly encouraging. The problem was that the dose that was required, 0.3 mg per kilogram, was just a too large a dose to be used in a clinical setting. And so in part for those reasons and the fact that lower doses didn't appear to be effective enough, the use of leptin for the treatment of obesity foundered and continues to some extent to founder. That's despite the fact that there are some clues that suggest that leptin might be usable as a therapeutic for obesity. I'd like to share some of that with you now. For one thing, it appeared that there was a responder subset. So while some patients responded a little bit and others not at all, there were some patients who responded actually quite robustly. So in these unpublished data provided to me by Alex DePaoli from Amgen, they showed that about 12% of patients receiving leptin showed a greater than 10% weight loss with 0 in the control group. That's actually a pretty significant weight loss, 10%. And moreover, 35% of obese patients on leptin lost more than 5% of their weight. So if one now begins to think about segmenting the obese population, there's certainly reason to think that there might be a responder subset. And if you think about it, knowing, of course, that leptin treatment of the leptin-deficient child is so effective, you might imagine that the responder subset could be enriched for if you selected obese patients who had relatively low leptin levels. I'll show you a slide of that in a moment. But first, I want to at least show you some evidence in a didactic form, at least that lends credence to the idea that patients with obesity and relatively low leptin levels would respond. First of all, as I mentioned, patients with absent leptin respond very robustly. But leptin also reduces weight in wild-type mice and in patients with low leptin levels for other reasons, including, as I'll tell you in a moment, lipodystrophy, but also a condition known as hypothalamic amenorrhea, subset of patients with NASH and others. In addition, there's published and unpublished data showing that leptin could reduce weight regain in patients who have lost weight on a very low-calorie diet. On that diet, leptin levels fall, and so leptin replacement has been shown to blunt that weight regain. So are there patients with low leptin levels out there and obesity, and might they be treatable if one could identify them by measuring leptin levels. Well, just yesterday, I got the following email, which I found pretty intriguing. This gentleman emailed me because he found out that he had a BMI of 33 and was obese with very low leptin levels of 1 nanogram per mil. He's being treated now with testosterone and thyroid treatment. I'll ask you to remember that in a few slides when we talk about some of the neuroendocrine effects of leptin deficiency. And he closed by saying he's never discovered the cause necessitating these treatments, and he wonders if, in fact, they're driven by leptin deficiency. Now, I think the sort of low leptin levels of this sort that are sometimes in obesity will be uncommon as a percentage, but as a total number, it actually could be significant, and this could be a case where a large number of patients could benefit by hormone replacement. And so that point is sort of made on this slide again, which I showed you earlier. If you look at any level of percent body fat, the range of leptin levels is quite broad. And so these patients here are quite obese, and several of them in this small cohort have leptin levels below 10 nanograms per mil. And I suspect if we broaden the cohort, you could even find patients with lower levels still, such as the gentleman who wrote me that email. So I think one strategy going forward might be at least to see, is there a subset with low leptin levels, and can we treat them more effectively with leptin? Then you might ask, what are we going to do about the patients who are clearly leptin resistant and for whom we don't imagine any more than a small fraction of patients would respond? Well, there are two things we could do. One is to try to see if we could find a leptin sensitizer, and the other is to try to understand more about the basic underpinnings of leptin resistance and develop new therapies based on that. As far as leptin sensitizers, I'll refer you simply to one paper. I won't talk about it in detail, but suggest that there's a lot of evidence in animals and even evidence in humans that certain agents can sensitize to leptin and that lead to weight loss as part of a combination therapy. And in this paper published in the PNAS in 2008, leptin was combined with another peptide hormone, amylin. Some of you may know this as Simlin or pramlintide, a therapy for type 1 and type 2 diabetes. It has some modest weight-reducing effects. But importantly, if you now combine leptin plus amylin, you get across-the-board dramatic decreases in weight of 13.9% in the subset of patients that was studied. Amylin has been shown in animals to resensitize to animals to leptin for reasons that aren't known, but at least this raises the possibility that leptin plus other agents might also be useful as a therapeutic, and that a deeper understanding of the physiologic mechanisms by which leptin and this other hormone act could provide new opportunities for the treatment of obesity. So as a summary of this then, leptin has potential as an obesity treatment either as a monotherapy for a subset of patients, probably low leptin obese patients, and as part of a combination of therapies including amylin or in other studies based on animal data, GLP-1, celastrol, and others. In the long term, however, we'd really like to understand what the basis of leptin resistance is and thus try to understand means for improving it. This will require studying a complex neural circuit that responds to leptin. We now know quite a bit about the neural populations on which leptin acts and that regulate appetite. On the left side of your slide, you'll see a pathway that says leptin receptor and NPY. This pathway normally acts to increase appetite. On the right, somewhat illegible, is a pathway that says leptin receptor and then MSH and MC4 receptor. And this pathway acts to decrease appetite. Leptin acts by inhibiting the green neurons and activating the red neurons. And the reason we know that these pathways are important for human physiology is that mutations in it also cause human obesity, and that includes not only leptin, which I showed you, but the leptin receptor, MSH, and the MC4 receptor. This is work done by others. In aggregate, these mutations and other mutations in the neural circuit that regulate appetite probably accounts now for 10 to 15% of morbid obesity. Morbid meaning BMI over 40. So that's a substantial genetic loading, a greater genetic loading than perhaps any other complex trait that's been described to date. And moreover, I think the number will grow with the advent of whole genome sequencing, which is being done in a number of groups around the world. Well, that's the story for obesity, but I want to now turn to the abnormalities that I mentioned the mice display, infertility, hypothermia, panendocrine abnormalities, and now tell you one other aspect of leptin physiology that devolves from that. Because in retrospect, it turns out the OB mice manifest that this constellation of symptoms, infertility, hypothermia, immune alterations, are abnormalities that are not typically seen with obesity. Rather, they're seen with starvation. And what that and other lines of evidence suggested is the following, that when fat mass is lost during, let's say, a period of starvation and leptin level falls, the low leptin level triggers what we might consider to be the adaptive response to starvation. This will include, of course, increased appetite and food intake if food is available, but also includes a whole host of other secondary changes, the aggregate effect of which is to reduce energy expenditure. And this includes the acquisition of a euthyroid 6 state, alterations of immune function, reduction of insulin signaling, the so-called insulin resistance of starvation, and decreased reproductive function in males and particularly females who develop hypothalamic amenorrhea in such circumstances. I'll point out to you from the letter I just shared with you, he reported being treated with thyroid and androgens. And so in some ways, he may be that young man may be manifesting some of the signs of leptin deficiency, not just at the level of increased weight, but also in this neuroendocrine axis. Now, the idea here is that when leptin levels are low, these responses are activated. When you give leptin back, the responses are abrogated. And this then led a number of endocrinologists, including Alex DePaoli, to consider the possibility that there might be pathologic states that have low leptin levels for one reason or another, and that if these patients with low leptin levels manifested some or all of these abnormalities, you might be able to treat them by giving leptin back. And the one example I'll tell you about today, there are others that I won't have time to tell you about, has to do with insulin signaling. So the question is, are there human conditions associated with low leptin levels and insulin resistance? And indeed there is. That condition is known as lipodystrophy. Lipodystrophy is essentially the fat equivalent of muscular dystrophy. For one or another reasons, often genetic, fat mass doesn't develop fully. Because there's less fat, there's less leptin, and these patients develop a severe metabolic disorder with severe insulin resistance, steatosis, and hyperlipidemia. So this is an example of that. This patient has generalized lipodystrophy. It can be caused by mutations in the genes that are shown below. This woman actually had, I believe, she had a gene mutation that caused lipodystrophy. You'll note before leptin, her body is covered with xanthomas, and she has a massively engorged fatty liver. After a year on leptin, no xanthomas, a normal-sized liver. So this is leptin replacement. And here are some of the parameters in this young woman. HbA1c fell from 13 to 5, no longer on insulin therapy. Her triglycerides went from 6,300 to 179. Urinary protein went down as well. Now, based on the response of this patient and others like her, leptin was approved in 2014 as a therapeutic. But I should emphasize that approval is only for what's called generalized lipodystrophy. It is not yet approved for partial lipodystrophy. Now, while the approval for partial lipodystrophy is in process, both in the EU now and in the United States. I'd like to point out that while general lipodystrophy has a very characteristic appearance as this woman exhibited, partial lipodystrophy does not. These three women, Andrea, Gail, and Mary, all of whom are working at an advocacy group for lipodystrophy, all have the condition, and yet none of them have an appearance that you would immediately pick out as being lipodystrophic. The response of leptin to leptin among partial patients is often very good, but variable. In general, the more severe the metabolic abnormalities in these patients, the more profound the response. We've been wondering for some number of years now, how does leptin improve the insulin resistance and the glucose metabolism of lipodystrophic patients? Now, the obvious explanation would be leptin suppresses food intake and reduces body weight, and that would be the explanation. But in a series of pair feeding experiments that Christina Hedbacker in the lab did, it became pretty clear that that was not the explanation, that some other mechanism was responsible for the effects of leptin on glucose metabolism. And in part for the reasons I shared with you, that leptin acts primarily on the brain, we considered the possibility that leptin's effects on glucose metabolism were mediated by modulating the activity of specific subpopulations of neurons in the hypothalamus. We did one physiologic experiment to test that, and that's shown on this slide. And our hypothesis was that ICV leptin would modulate the activity of glucose-sensing neurons in hypothalamus. So to begin to test this, Sarah Stanley in my lab, formerly of Cambridge, where she worked with Steve Bloom, made a new mouse, and this mouse inserted the Cre recombinase into the glucokinase locus as a means for directing Cre recombinase to glucose-sensing neurons. Glucokinase is a key regulatory step in glucose sensing. And so by marking these neurons with Cre, we could study specifically what we believe to be glucose-sensing neurons. Here's a picture of that Cre mouse made it to a GFP reporter. The GFP is expressed in what we were able to show are glucose-sensing neurons in a number of hypothalamic nuclei, including the arcuate nucleus, the VMH, the dorsomedial nucleus, paraventral PVN, as well as the LH. So we now have an entry point with which to study the function of specific glucose-sensing neurons and establish their relationship to leptin signaling. I won't show you the data, but I'll just tell you that many of these neurons also respond to leptin. So there's some processing of both glucose and leptin information by these neurons, especially in the region I'm going to tell you about, the VMH. Now, at this point, we wanted to see what happened if we control the activity of these neurons remotely, and we might have turned our attention to optogenetics, and I'll show you one optogenetic experiment in a moment. This is a means for controlling neural activity with light. But Sarah and I decided that we would like to try to develop another means that, for reasons I'll come to, we thought could have more therapeutic potential in human. And we asked ourselves, is there a way to control neural activity remotely without the requirement of a fiber optic cable, which is necessary for optogenetics? You have to fire a light to activate this light-activated channel. We wanted to see if we could develop an analogous means, but that made use of a radio wave or a magnet. And initially, we chose a radio wave frequency in the AM band, 454 kilohertz, and that's because 465 kilohertz, because that radio wave frequency is already used in clinical medicine to control pacemakers, cochlear implants, and so on. So the first thing we needed was a radio wave generator. Turns out radio waves are used in commercial welding. It's so-called dry welding. If you take two pieces of a pipe and you oppose them together in an electromagnetic field, the metal heats as it would in a microwave and fuses. That's dry welding. So we called a group of dry welders, and to their and our amazement, they came over with a radio wave commercial welding device that we used for the first set of experiments I'll tell you about. What you can see here is there's a coil, and inside the coil, a radio wave field is generated. So how are we going to transduce a radio wave into a neural signal? Well, we went through a series of iterations of this, but the system we ultimately developed is a two-component system. The first is a channel known as TRPV1. TRPV1 is a heat-sensitive channel. It senses thermal pain. It's the channel that gates when you eat a hot piece of pizza and recoil. It's also the target for capsaicin or chili pepper, and the reason that capsaicin feels hot is that it's chemically gating a channel that's normally opened by thermal pain. The second component is ferritin. Every cell in the body uses iron, but iron unsequestered can be toxic. Ferritin is an iron-binding protein that spontaneously forms a protein shell surrounding a 5 to 10 nanometer iron oxide iron nanoparticle. And our original idea was something like this, went like this. We can use a trick to tether the ferritin to the channel. The radio wave would heat the metal, and the channel would open. Ions would flow in, and there'd be electrical changes in the cells that are treated in this manner. So that was the idea, and indeed it worked in vitro much better than we thought it would. And so rather than go through all those in vitro data, I'll just advance immediately to the in vivo data, where we now use this to try to control neural activity. So what's done here is that the constructs are going to be injected into the ventromedial hypothalamus, the VMN. And what Sarah does is she makes a virus that's in the off configuration that expresses both TRPV1 and tethered ferritin. She injects it into the glucokinase cree mice and now fires the radio wave and asks what happens. And here's the result. In blue, you'll see a similar set of experiments using the more commonly used method of optogenetics. That's in blue. Green is using the radio wave, and then black is the control. And what you can see here is if you activate these glucokinase-expressing neurons in the ventromedial hypothalamus, in both cases, you see about a 75% increase in blood glucose. And the total efficacy is the same for the radio and the optogenetic methods, although it might be a slightly slower onset for radiogenetics. But the overall effect is the same. Activating these neurons causes a massive increase in blood glucose. Why is that happening? Well, glucagon has now tripled and insulin has fallen. Well, if blood glucose is going up and glucagon has gone up, it would seem to mean that it's the glucagon driving the blood glucose increase. And glucagon generally rises in a state of hypoglycemia. In fact, it's mediated by sets of what are referred to as glucose-inhibited neurons. And so the idea here is that normally when glucose is low, these neurons are on, glucagon would be high and insulin is off. We did our experiments in fed animals when these neurons would normally be off. But if you artificially activate these neurons using the radio wave or the light, you get what you might consider to be the response to hypoglycemia, and glucose level rises as a consequence of an increase of glucagon. So in a sense, I'm saying that these neurons are components of the response to hypoglycemia. Okay, we now wanted to ask what happens if you inhibit these neurons. And to do this, Sarah reasoned that we might be able to make mutations in the TRPV1 channel that convert it to a chloride channel. That's shown schematically here. It turns out that two other members of this family have similar structure, and mutations in the pore domain had been successfully used to convert them into chloride channels. So Sarah introduced the analogous mutation into this channel and then showed in vitro using chloride imaging that indeed this was now a chloride channel, as shown by this quenching of the chloride fluor. So the conclusion here is a point mutation in the pore region converts TRPV1 into a chloride channel. What happens now if we inhibit rather than, as I showed you before, activate these neurons? Well, now blood glucose falls in half. Dramatic decrease. The only difference between the two experiments is the form of the channel we use. And why did glucose decrease? Plasma insulin tripled, while glucagon failed to rise despite the fact that the animal was hypoglycemic. So we got essentially the opposite effect. Now, the fact that we get opposite effects depending on which version of the channel we use tells us that this isn't an artifact that's dependent on the channel, and we think it validates the method as well as having some physiologic implications I'll tell you about. Okay, so I want to digress for a moment now and at least tell you about our expanded view about why this even works. Now, originally we used TRPV1 and we thought that it was working simply by heat. We'd heat the particle and gate the temperature-sensitive channel. That simple possibility, however, was negated when we then went on and tested a temperature-insensitive version of TRPV1, and that still worked. So we think temperature can contribute, but the method can work via other means besides a temperature transfer. And we've since considered the possibilities of mechanotransduction and/or a local voltage change, inducing an energy transfer between the particle and the channel. Now, I won't go through the theory behind each of these and why we think they both might contribute. Rather, I just want to show you, to tell you that as part of considerations the mechanotransduction could be relevant, we started using a magnet. Ferritin is paramagnetic, so if you apply a magnetic field, the magnetic moments will align and exert a magnetic force between adjacent particles. So as an empiric exercise, all we're doing here now is exposing cells or animals to a magnet and asking, do we get a similar response to what we saw for radio waves? And here's the response using simply a handheld magnet on a micromanipulator in neural slices. In this panel, what you see is if you apply the magnet to the wild-type channel, there's a massive increase in firing rate. If you do the same thing for the mutant channel, there's a hyperpolarization and a cessation of firing. So the magnet works, And this simply summarizes the effects of either wild-type or mutant channel on membrane potential and firing rate. Well, we wanted to then use this information in physiologic studies to ask if a magnet can be used to control behavior. So I told you before that we thought we were working with glucose-inhibited neurons. And you'll note, of course, that when an animal is made hypoglycemic, it will eat more. So now what we wanted to do is take fed animals and see if activating these neurons would now increase food intake. So we did the following experiment. We injected the animals, as I showed you before. The conditional virus is injected into the VMH. Animals are fasted for 24 hours, and then they're placed either far away from or in proximity to a standard MRI machine. And we record food intake during 3 intervals after a fast. In period 1, when the animals generally eat the most, they're placed far away from the field. In period 2, when they eat a slightly lower amount, they're placed close to the MRI machine. And in period 3, they're moved back far away from the field. And the key interval here is interval 2. Does exposure to the radio wave field, the magnetic field, increase food intake in period 2? Indeed, it does. That's shown here. These are the control animals in the lighter shade. You see a progressive decrease in food intake over each of the 3 intervals. But in period 2, you'll note a tripling of food intake that's statistically significant. So activating these neurons can certainly drive food intake. What about the opposite? Well, in this case, we took a fasted animal that would normally be eating a lot, and we inhibit these neurons, and now we ask, does it decrease feeding? So it's the same experimental design, except now they're put in the high magnetic field only in period 1, immediately after the fast. And here's the result. In period 1, there's a dramatic decrease in food intake with what looks to be a compensatory increase in food intake. So in this case, the experimental group is in the lighter shade. So you see a decrease in period 1 and now a compensatory increase in period 2. So this tells us that these neurons play a role not only in regulating glucose metabolism and endocrine hormones, but also integrate behavioral responses with them, regulating each of the features of the response to hypoglycemia, but also the response to hyperglycemia, as evidenced by the results of neural inhibition. So among other things, this has changed my view of how blood glucose is regulated, because I think the data would suggest that the CNS is perhaps the prime or principal site at which blood glucose is sensed. I should mention there's a lot of experimental support from this, including the work of Abraham and Bloom, as well as Lott and Vrodnich. But I believe that the brain is the primary glucose sensing organ, and that we can consider the pancreas, in a sense, as an effector organ for CNS circuits that sense glucose, so that brain neurons secondarily control glucagon and insulin levels. Why would this make sense? Well, for one thing, the brain is the primary consumer of glucose, and so it would make sense that it be the primary driver of blood glucose changes. Why then, you might ask, do we have glucose sensing capability in the pancreas? Well, this is a critical requirement for living organisms to maintain glucose homeostasis. And so you certainly don't want so you have glucose sensing in the pancreas as a fallback in case the brain systems are damaged, in part perhaps by hypoglycemia. So I think it's reasonable to propose that islet hormones function in many cases as an effector limb downstream of glucose-sensitive CNS circuits. Now, I'll close by just saying that apart from the physiologic implications, we'd like to believe this method may have broader utilization. Sarah is now knocking in the TRPV1 ferritin constructs into the mouse germline, and this could allow non-invasive activation or inhibition of dispersed or localized cells, even perhaps in inaccessible sites or during development. We're trying to make simple devices based on wireless charging coils to begin to make it possible to perhaps just breed 2 different animal strains and use a magnetic field to control cellular activity. We also think there could be applications of this in human. A number of human disorders are treated with deep brain stimulation. And so could you imagine using a method like this as an alternative of DBS for Parkinson's? What about Prader-Willi syndrome, where bariatric surgery is ineffective, or even as an alternative to bariatric surgery? So I think it's fanciful to get too far ahead of ourselves here, but we did at least one experiment to see if this method could be used in a region-specific manner to control metabolism. And in this experiment, what Sarah did was to inject the activating versions of the construct directly into the LH, lateral hypothalamus. There's no conditional expression at all, just all the regions, the neurons in the regions would express it. And here's the result of activation there. You can see a marked decrease in food intake and also a decrease in blood glucose. So in this case, the magnetic field can reduce food intake and glucose in a tunable manner after expression there. And so we're beginning to think of whether or not there are certain indications that might be amenable to the use of this as an alternative to the highly invasive method of deep brain stimulation. Okay, so I'll close here, and I'd just like to summarize by reviewing what I hope to have communicated today. The cloning of the OB gene and identification of leptin has uncovered a new endocrine system regulating weight. The system provides a means by which changes in nutritional state regulate maybe all other physiologic systems. There are several leptin deficiency syndromes that are treatable with leptin replacement. This includes lipodystrophy, an underdiagnosed cause of diabetes that is treatable with leptin. A substantial fraction of morbid obesity is the result of Mendelian defects in the neural circuit that's modulated by leptin. A subset of obese individuals lose weight with leptin therapy. The majority are leptin resistant but may respond to a leptin-amylin combination. And that we developed a nanoparticle-based method to show that the CNS can control glucose metabolism by regulating hormone secretion by pancreatic islets. And at a high level, I would have hoped to have at least encouraged you to consider the possibility that obesity is a physiologic endocrine disorder. So I want to just close now by acknowledging my colleagues. The original work on leptin and glucose metabolism was done by Seika Kamahara, Ezra Asselmas, Christina Hedbacker, and Sarah. And the nicer work I told you about, the nanoparticle work, was done by Sarah Stanley, Leah Kelly, John Dordick, and Jeremy Sauer, our collaborators. So thank you again for giving me the chance to speak today, and thank you.